Hello viewers, welcome to this particular lecture on microbiology. In this particular lecture, I shall be talking about the cultivation technique of edible mushroom. Now mushrooms belong to the fungal kingdom. Actually, they represent higher fungi commonly called bracket fungi. And the unit of this bracket fungi actually is represented by a thread-like structure called mycelia, which gets interwoven, producing different types of tissue, which is commonly called the pseudoparenchyma or plectenchyma or prosenchyma. And mushrooms actually can be classified as edible, which I will be talking about today, but they can also be non-edible. Non-edible meaning they may cause minor toxicity, and thirdly, most importantly, they may cause death and they are deadly poisonous mushrooms. Now normally, this particular mushrooms can belong to two specific classes. They are ascomycetous fungi and basidiomycetous fungi. And normally they will reproduce by producing spores, or at the same time the threads will undergo fusion. But normally two haploid cells undergoing fusion will produce an intermediate stage called N+N stage, which ultimately if they are compatible they will meet, otherwise they separate. But the mushroom fruit body or fructification actually is the spore bearing structure, and this particular structure may be colorful. At the same time they may contain high amount of protein, and the different parts of this particular sporocarp may be a typical stalk or stipe. There may be the upper part with the pileus. Just below the pileus there may be the gills containing the spores, which can be ascospores or basidiospores, and at the same time there may be the veil-like structure which is actually covering the young sporocarp, and it breaks open when the sporocarp gets matured. Now mushrooms actually can be cultivated or it can be collected from natural habitat. This particular sporocarp of Agaricus bisporus is a cultivated button mushroom. So it is having a global palatability, consumed all over the world. But at the same time, this particular ascomycetus morchella is not cultivated because it cannot be cultivated. It is growing along with some higher plants, growing in a form of a symbiotic association called mycorrhizal association. So that is the reason why this particular mushroom has got a high palatability, and at the same time it is very costly because it cannot be cultivated. Now there may be different types of mushroom. These mushrooms can vary in their size, in their shape, in their color. There may be the pleurotus, there may be the colibia, there may be the cantharellus, there may be the clitocybe, there may be the tricoloma. You can very well see that they vary in their color, and there may be the rusula, or sometimes there may be the hygrocybe, amanita, laccaria, lepiota. All these are different forms of edible mushroom growing in different natural habitat, and they also grow in different types of substrate. Sometimes it can be paddy straw, sometimes it can be wheat straw, sometimes it can be grass, sometimes it can be root of higher plants, sometimes it can be logs. So different types of mushrooms are there, and they grow in different environmental condition from tropical to temperate or even subtemperate climate. Now let us consider certain mushrooms which are harvested from their wild habitat. Some of these mushrooms include the typical tricoloma. This tricoloma magnivelare is actually a white fruit body growing in humus soil. But when we consider this typical cantharellus, cantharellus is actually an oyster mushroom because it looks like an oyster, but it has a typical orange pigment or carotene pigment, and that is why this color. There may be the wild sporocarp of craterellus. Now this craterellus actually is typically purple in color because it has anthocyanin pigment, and at the same time they may be horn-like, horny projections. So sometimes commonly they are called horn of plenty. But when we come to Boletus, this is a unique mushroom where the stipe is thick but the pileus is somewhat brown in color. But this mushroom actually grows in the pinus root. It grows along with the pinus root as an ectomycorrhiza. So wherever there is the pinus plant, this particular mushroom will grow. There may be the sporocarps of tuber gibbosum. It grows underground, and there may be specially trained sniffer dog who finds out this tuber because it is having high value in the market of food technology. There may be the hydnum repandum, which is also a very important mushroom, which is commonly called the spreading hedgehog because of its unique habitat below the grass, and it is having a typical dented gills. There may be the wild sporocarp of Hericium. Hericium actually is commonly called the coral tooth because of its dentate appearance. Or when we come to Lepiota rhacodis, Lepiota is having a big trunk like stipe which is white and a typical umbrella shaped pileus with multiple scales on top of it, which are nothing but basically the fragments of the vulva. We also have typically this black picoa, or it is the Picoa carthusiana, which is having typically potato like structure, and it also grows underground and it has high amount of carbohydrate. But when it comes to the wild sporocarp of Sporacis, this particular Sporacis is a typical cauliflower mushroom because it looks like a cauliflower. So you can very well see that the variation in the sporocarp structure and this variation and color and texture and shape and size makes mushroom a unique feature. Of course, Pleurotus, which is a very common oyster mushroom, grows in multi-tier. It can be cultivated, but it can also grow in tree trunk, which is also a very palatable mushroom commonly consumed in the Southeast Asian countries. But when we talk about natural harvest, there may be some disadvantages, which include their availability is not regular. So naturally the prices go up and down, or sometimes there may be some toxicity involved in it. We are not sure whether there is any toxic implications or not. And particularly there may be some conflict because there are collectors who collect these mushrooms. So there may be conflicts among the collectors, and sometimes in big national park these mushrooms grow, and when you are collecting these mushrooms, the authority will prevent you. They will make you pay some levy when you are collecting it because these are costly things. And we should keep in mind that people might think that when we collect the mushroom we are harming the biodiversity, but not really, because when we take up that sporocarp, the mycelia is already there below the ground. So this mycelia will again grow and will again produce a new sporocarp. So it is actually not a problem with respect to destruction of habitat or destruction of biodiversity when we pick up the mushroom. But at the same time, of course, we must say that mushrooms are very delicate organisms. Whenever we are picking it up, sometimes it so happens that the growth rate might be reduced or stopped. So it is better that if we can cultivate these mushrooms in a natural habitat, but some component of artificiality is induced. Like when we grow these mushrooms from or on logs, what we do is, these are the logs which are arranged in crisscross arrangement. Say for example, a specific size of these logs are taken and say 3-inch diameter hole is made, and they are actually the mushroom spawn, which is the seed of the mushroom, which is added to it. And then these mushrooms will grow in it, and you can see here how these mushrooms have grown. This common mushroom is called shiitake and the scientific name is Lentinus edodis. So this is how we say that we have grown this particular mushroom in external habitat, but utilizing the artificially grown spawn. So it is called partially external or partially controlled cultivation of edible mushroom. But when we talk about cultivation, we always do it internally. So it is called internal tray cultivation, which is popular globally, and at the same time it is popular in different households because it is grown very easily. You can grow it very easily. So when we talk about indoor cultivation, mushroom cultivation may have different phases, and these phases include composting, include spawning, include casing, appearance of pinhead and harvesting. These are the major stages. Now let us consider each of these phases one by one. Composting is commonly preparation of the substrate. How we prepare the substrate, it can be done between 40 to 60 degrees centigrade. It should have a regulated amount of carbon and nitrogen, and there may be some additives given. Ultimately, this particular substrate is composted using bacteria or using some other fungi so that the substrate is readily usable by another fungi or another edible mushroom. So this is how we do the composting, mixing of this particular mixed compost, turning it and making it suitable for the fungi. But composting is followed sometimes in a mechanized manner. So this is the typical machine which is called mechanized composting, by which we can use a motor so that this churning is done automatically. And when we talk about the next phase, that is the pasteurization, which is a very important step. We will apply hot water, say at elevated temperature, maybe 70 to 80 degrees centigrade. By this, we will kill the pathogenic bacteria which will prevent the growth of the mushroom through the production of antibiotic. So this mushroom substrate is ready and now it will be inoculated. And this phase commonly includes two steps. One is the spawning, another one is the casing. Now what is spawning? Preparation of the seed of the mushroom, which is actually wheat grain commonly used, which is mixed with calcium sulfate and calcium carbonate in specific percentage, inoculated with the mushroom mycelia, and then it will grow. It will grow in that bottle or in that packet, and it will mature as a white mass, thick mass. And this is how it looks like. This is how the inoculation is done, and this is how it looks like after the spawn gets matured. But these are wheat grains actually. And when they get matured, this is the tray where they are being matured, and usually spawn growth is better when we store it in darkness. And this is how the matured spawn looks like. And then comes the most important step, which is casing. Actually, in casing what we do is we apply a typical layer of soil and sand together, and thereby what we do is we give a shock. A shock meaning there is no contact with the air. There will be enrichment of carbon dioxide, and this condition actually enables the mycelia to grow into sporocarp. Why? Because sporocarp means sexual reproduction, and fungi has reached the end of its life cycle, so it will produce the sporocarp to produce sexual spores. So that is why casing is a very important step by which the sporocarp induction is actually done. And after that, what is there? There is the pinning or appearance of pinhead, and these pinheads are nothing but the young mushrooms, and the cropping or harvesting meaning the picking of these mushrooms. These are the pinheads, how they appear. You can see here a very unique point. Usually the stalk is fibrous, but the top, the pileus, is what we consume because it is fleshy, it is palatable. So naturally, whenever there is too much carbon dioxide, the growth is actually hampered. What growth? The growth of the pileus. So the size of these mushrooms may vary depending upon the availability of oxygen. And actually these mushrooms may be grown in multiple tier, and this is how they are grown and harvested from this bed. Mind you, these are the button mushrooms which are grown in a big farm. You can see they are just like flower bed. But when they are picked up, it should be done carefully with a mechanized device so that the base of the mushroom is not damaged. If it is damaged, then naturally production will be hampered, because it is not that this bed will produce the mushroom only for one time. Actually there are multiple flushes, say first flush, second flush and third flush. At least 3 flushes are given by a bed, but every time after every flush the amount of mushroom sporocarps is getting reduced. So the very important point is, what are the components that we use for the substrate, and at the same time what is the condition of this substrate. So I have already told you that the substrate should be locally available, the substrate should be cheap, the substrate should be enriched in carbon and nitrogen. So this type of substrate may include the sawdust, may include the straw, may include the corn cob, maybe coffee pulp, maybe seed hulk, maybe cotton seed meal, or maybe soybean meal. Different types of substrates may be used, but at the same time there may be some additive of nitrogen in the form of urea, in the form of ammonium nitrate, in the form of animal dung, animal manure. Mind you, when we are using it, it should be partially pasteurized or sterilized, otherwise there will be growth of some excessive bacteria which will prevent the mushroom to grow. Now coming to some important commercial species which actually can be cultivated globally. Some of these species may be natural or may be artificial, but here this particular agaricus, you can see this agaricus can be grown anywhere. Particularly in India, it is grown in the foothill region of the Himalayas where the temperature is very low. In West Bengal, it can be grown in the northern part because the temperature is low there, and they require a temperature of anything between 15 to 20 degrees centigrade maximum. But when we consider this auricularia, it is a typical slimy sporocarp. So it is also called the slimy fungus, also called the jelly fungus, also called a typical name devil's ear, and they are having some particular medicinal importance also, though the palatability is relatively low. There may be the Flammulina velutipes, which is also very important because it has high carotene content, so vitamin A production will increase. So these are the natural velutipes or Flammulina velutipes, and these are the artificial ones. You can see clearly that when they are grown in artificial condition, the production of this carotene pigment has been lowered. So this is what is happening when we are growing a particular mushroom in artificial condition, the pigmentation may be varying. Or this is typically Hypsizygous tessellatus. When it is grown in artificial substrate, the size of the sporocarp may be reduced. There may be slight pigmentation at the top, but when it is grown in natural logs, they are much bigger, and naturally it is said that the palatability has also increased. This is typically the lentinus. You can clearly see the difference. This is artificial cultivation. It varies in size. Artificial cultivation, the sporocarps have been reduced, and in natural cultivation, the sporocarps size have been increased, though the production is relatively less, and at the same time the number of scales also vary. Perhaps they look like ice cream cone, very attractive. But when it is pleurotus, pleurotus actually is a typical oyster mushroom, and it can be grown anywhere within the household. Only it requires a relatively darker place, a shady place where moisture content should be high. So when it is grown artificially, it will be produced in multiple tiers. But when it is grown in natural habitat, the size may be bigger, but the tire size becomes smaller. This is another important mushroom in rural Bengal. These are called poal chhatu basically. They are actually called Volvariella volvacea. Why is so called? Because the vulva or the veil is covering the entire mushroom, and this particular mushroom at their egg stage is particularly palatable. And this is the point where we harvest it. But when they are grown in natural condition, when they are growing in this condition, they will grow into a massive sporocarp and they will spread like an umbrella. But unfortunately, in this condition, the palatability is reduced. Now, let us discuss some major pests or harmful organisms that reduce mushroom production. One important pest is the maggots. Actually, the larvae of insects, they grow in different plants including paddy. So when we are using paddy straw substrate, when we are using casing soil, if they are not properly sterilized, then they will come. They will feed on the mushroom. How will we understand? There will be holes at different points. So thereby we can say these mushrooms are destroyed by insect pest, and once they are destroyed, naturally the productivity is low, the flush content is reduced. There may be sometimes infection of bacteria. These are pseudomonas growing inside mushroom tissue, inside the pileus, and these bacteria, they do produce certain antibiotics, and these antibiotics will reduce mushroom growth, reduce production, reduce the fruit body size. Not only that, there may be certain bacteria which will colonize on a particular fruit body and actually will create a typical blotch-like disease. What is blotch-like disease? There will be wet surface on the white sporocarp. So naturally you can very well understand, when we are looking for a milk white mushroom, if there is some blotch or if there is some dark spots in it, we will not select it. We will say, okay, I will not consume it. So basically friends, what we have learnt is, there may be different types of mushrooms which can be collected from natural habitat, which can be cultivated artificially with different stages. We can keep a strict vigil on the cultivation. We can produce tons and tons of edible mushrooms, but we should be definite that this particular mushroom is without any contamination of bacteria, or it doesn't contain any toxin, or it doesn't contain any heavy metal. Why heavy metal? Because mushrooms are growing on the soil, mushrooms are growing on a substrate where if the metal is present, then the metal will be taken up by the mushroom, and in this way it will come to the food chain. So mushroom consumption is good, but there is a word of caution that you should consume from a registered farm with the FAO license, and naturally that will be definitely safe for you, and definitely it will do good to your health as a vegetable source of protein.